Greetings, everyone. Welcome to the Science of Childhood Cancer lecture series hosted by the St. Jude Comprehensive Cancer Center. I'm Martine Roussel, member of the Department of Tumor Cell Biology. And today it's my great pleasure to welcome our speaker, Dr. Yael Mossi. So Yael is not a stranger to many of you. She was here at St. Jude to give a seminar 5 years ago. I met her when she came to St. Jude. And recently, Yael and I have been collaborating on a Crazy 8 initiative project drugging the undruggable MICAN to treat MICAN-driven medulloblastoma and neuroblastoma, for which he's a PI. So before I formally introduce Dr. Mossi, I'd like to note that you can submit questions at any time during the talk using the chat function and sending questions to all panelists. So Dr. Mossi also kindly offered to stay on for some additional Q&A after the top of the hour, if anyone would like to stay for further discussion. So now I'm delighted to introduce Dr. Mossi. Yael obtained her MD in Tel Aviv in Israel, after which she moved to Philadelphia to do her internship in pediatrics and her postdoctoral fellowship at the Children's Hospital of Philadelphia, where she is now an attending physician, Associate Professor of Pediatrics with tenure at the University of Pennsylvania School of Medicine. Yael is well known for her work in understanding the hereditary predisposition and progression of neuroblastoma. She discovered that the region of chromosome 2 was associated with neuroblastoma, and then she proceeded in identifying the oncogene ALK as a driver of the disease. And then she showed that it could either be genetically inherited or acquired. As you may know, many ALK inhibitors are in the clinic, and she and her team are translating these drugs for the treatment of neuroblastoma, and I'm sure that she will present this work today. So on a personal note, Yael Mossi has already received many honors. She's a member of the American Society of Clinical Investigation and the American Pediatric Society, to cite a few. What's very impressive about Yael is that she has a dazzling number of committees that she attends, and she manages everything. She treats children with neuroblastoma. She consults for Roche Genentech for their pediatric oncology drug development, Pfizer, and other pharma. She's a member of the Children Oncology Group. And she's the chair of a COG phase 1/2 clinical trials. She's on study section, and she's a mother, and she has a wonderful family. So without taking more of your time, Yael, we're looking forward to your presentation.
Thank you, Martine, for that really kind introduction. And I'd also really like to thank Charles for the invitation. It's truly an honor for me to be speaking in this Science of Childhood Cancer lecture series. It's actually my clinic day today. And so it's really a unique way to balance science and clinical practice. And I feel it's my privilege to be able to sit at the intersection of doing both of those and integrating them. And so the problem that all of us really think about and are tackling is that progress in curing childhood cancer really has plateaued. And I'm just going to, move this bar up here so I can see my apologies. All right. So progress in childhood cancer really has plateaued, and this is particularly true for children with solid tumors. And as you see on the x-axis, this only goes to 2005, but really this could be extended out to almost 2022, where despite maximal augmentation in the intensity of our therapy, there's been a plateau in our cure rates, and these have remained stagnant. And our therapies still largely remain empiric, especially for patients with solid tumors. The disease that we study in the lab is neuroblastoma, and I don't need to give much of an introduction to this disease, but it is thought to be due to a misappropriation of normal autonomic neurodevelopmental pathways, We know that the median age of diagnosis is quite young at 17 months, and it really does remain an important pediatric problem as it is responsible for 12% of childhood cancer mortality. And there are some known oncogenic drivers, the most common of which is McN, and I'll address that at the end, as Martine alluded to in her introduction. I'll talk a lot about ALK, and also ATRX, the RAS-MAP kinase pathway, PTPN11, and how that's involved and how we're trying to target this pathway. And then the core regulatory circuitry is, of course, really important in the lineage specification of how we how these neuroblasts eventually stop their maturation process and cause the disease neuroblastoma. And it's important to note that there are actually 3 FDA-approved drugs for neuroblastoma. They're all derivations of the same target of GD, which is GD2, and they're all monoclonal antibodies targeting GD2. We can cure about 50% of these kids, but when these children suffer a relapse, it remains a major problem. And still today in 2022, there are no known curative therapies. And these curves show the progression-free survival in red and the overall survival from time to enrollment on several phase 1 clinical trials for patients with high-risk neuroblastoma who suffer a relapse. So you can see that essentially it remains lethal, although with our improved understanding of the underlying biology of this disease, we have been able to extend life with quality, with more rational therapies. And our goal, of course, is to prevent relapse. So when I started working in the laboratory in 2000, my very first formal research project was to find the familial neuroblastoma gene. And I'll just mention that I had absolutely no research background and could barely even hold a pipette. What I found really interesting was that it was first described in 1967, actually, by Jane Chatton, who was a pathologist in Philadelphia at CHOP. And she made the observation of a child who had multiple disorders, including neuroblastoma, in 4 other siblings in the family. So this was in 1967 when this was first published in the New England Journal of Medicine. And then in 1972, Al Knudsen, who was a close friend towards the end of his life, but also a mentor of mine, in collaboration with Louise Strong, made the mathematical prediction that, like all other human cancers, neuroblastoma would have a hereditary component. He and Louise predicted that in the hereditary cases, one event occurs in the germinal cells and is inherited, and then the second occurs in somatic cells, kind of the classic 2-hit hypothesis. And he predicted that hereditary cases would account for 20 to 25% of all neuroblastomas. Now, at the time, we knew that there was a family history of neuroblastoma in a very small number of cases. 1.2% was what was in the literature. And these families have the classic features of a cancer predisposition syndrome, where there is an earlier age of onset of the disease. Often these kids have multiple primary tumors. There's incomplete penetrance, and the high mortality has made it very difficult to study, unlike diseases like breast cancer, prostate cancer, or colon cancer. In 2008, after collecting a number of 3-generation families where neuroblastoma was segregating in the family. And with the advancement of technology, we performed a genome-wide scan looking for evidence of linkage. And we found a signal on the short arm of chromosome 2, shown here, with a LOD score, a logarithm of the odd score of 4.23, really strongly suggesting to us that it was in this region where we would find the neuroblastoma predisposition gene. And of all the families that we had one in particular where the grandmother was affected with the disease. She had a daughter who was unaffected, but had to be the obligate carrier because it turned out that she had 3 children with neuroblastoma. And they were the only family where we could identify breakpoints, both proximal and distal, allowing us to narrow this region on the short arm of chromosome 2. And you can see that here at the telomeric end of the region is MYC-N, and all the way down at the centromeric end was ALK. And in between here were 104 genes. And so we started sequencing these using Sanger technology one by one and started off with MYC-N. I will say at the time, my mentor, John Maris, was sure that MYC-N had to be the neuroblastoma predisposition gene. And it was really thanks to families like this that we were successful in ultimately identifying germline point mutations in the kinase domain of ALK. I was contacted by this young woman who at the time was 16 years old. We had a protocol posted on the NCI website for the rare families where neuroblastoma is inherited. And she reached out to me and told me that she had a sister who died of neuroblastoma. She had another sister who had just been diagnosed with neuroblastoma, and she had a brother who had kind of a lazy eye, as she described it. So she gave me the privilege of attending one of their family picnics where we literally drew the blood of as many individuals as we possibly could, brought the blood back to our lab, extracted the DNA, and eventually this family really was key to our discovery of point mutations in ALK that lie in the kinase domain. And it turned out that the young woman who brought this family to our attention was a silent carrier. The interesting, story as she grew up is that she went on to wanting to have her own biological children and did have prenatal genetic testing and does have a child who did not inherit the mutant allele. So this family really was integral, and these data, along with 3 other groups, was published back in 2008, the discovery of ALK as the major familial neuroblastoma gene. And I won't belabor these pedigrees because this was published quite some time ago, but I do want to spend a little bit of time really talking about the history of the ALK gene. And it has a special meaning for me, especially today, as I have the privilege to talk with all of you, because as I think many or most or all of you probably know, the ALK gene was originally cloned at St. Jude, and it was cloned by Tom Look, when Steve Morris was a postdoc working in Tom Look's lab when he was at St. Jude, and this was in 1994, published in Science. And the gene was cloned in a cell line derived from a patient who had anaplastic large cell lymphoma. And Tom tells the really incredible story of knocking at the door of several big pharmaceutical companies saying that they really needed to develop a drug against this oncoprotein. He actually says that he brought all of these individuals from different pharmaceutical companies together at the boardroom at St. Jude, and they all seemed excited. And then when they left, nobody felt compelled to invest in developing a drug for an orphan disease. Since the discovery of ALK in 1994, it was initially thought to be an orphan, have an orphan receptor, but the ligand has since been discovered by 2 groups simultaneously. One done by Yossi Schlesinger at Yale, and the other with Ruth Palmer in Sweden. And we now refer to this as either FAM150 or augmenter. It has many names. It's been well known that a subset of inflammatory myofibroblastic tumors harbor ALK fusions, which are very different than what we're going to talk about when we speak of ALK point mutations in the kinase domain for neuroblastoma. And in fact, the presence of an ALK fusion in the context of an IMT can be detected simply by doing immunohistochemistry. And when IHC is positive for native ALK expression, that is a surrogate for an underlying ALK fusion. And then Christine Lovely published a beautiful paper in 2014 doing next generation sequencing and actually showing that up to 70% of IMTs harbor ALK fusions, and that a lot of these aren't necessarily found just by IHC alone. And so I show you here, a pictorial sort of schema of the ALK receptor with the transmembrane domain here, and in the majority of tumors where there are ALK fusions, the cleavage happens right here at the cell surface. And so there is no expression of the extracellular domain. Whereas in neuroblastoma, the receptor remains intact and the mutations lie intracellularly and are missense mutations in the kinase domain. It wasn't until 2007 when the Japanese group led by Dr. Soda and his team published in Nature that around 3 to 5% of patients with non-small cell lung cancer harbor an ALK fusion. And canonically, the partner is EML4. And I think that's really interesting because this really differs from the disease IMT, where their ALK is quite promiscuous and tends to have multiple different partners. And I think that is very relevant to the underlying biology as to why patients with IMTs have differential responses to ALK inhibition. And also important to note that for patients with anaplastic large cell lymphoma, especially pediatric patients, more than 90% harbor an ALK fusion. Almost always, the partner is the nucleophosmin gene located on chromosome 5, as first described here by Tom and his group. And this is really a purely oncogene-driven disease with very minimal heterogeneity, which is why ALK inhibition has had such a impactful and durable effect for these patients. Our group was the first to describe mutations and amplifications in neuroblastoma. These were really unique to neuroblastoma until these became discovered, especially the mutations shown here in the kinase domain, as mechanisms of resistance to ALK inhibition in patients with non-small cell lung cancer and inflammatory myofibroblast tumors receiving ALK inhibition. And since then, we now know that as we look for, and do more sequencing and do more RNA sequencing, we now know that ALK fusions can be found in subsets of renal cell carcinoma, thyroid cancers, Langerhans cell histiocytosis, and high-grade gliomas. So we took a step back and worked with the COG to sequence close to 2,000 tumors from patients at the time of diagnosis. These tumors represent the spectrum of the disease that we see in the clinic. So patients ranging all the way from low, intermediate to high-risk disease. And we found, using Sanger sequencing still back in the day, that the functional mutations and those that are disease causal all are within the kinase domain of ALK. Shown right here, there are in fact 3 hot spots. And these represent 85% of all mutations in ALK, specifically for patients with neuroblastoma. But there are also a number of these low frequency variants that I'll talk a little bit more about later. Now, mutually exclusive from the presence of an ALK point mutation, ALK kinase domain point mutation, we can see here on this SNP array that 4% of patients have high level ALK amplification. And it always co-occurs with MYC amplification, but they're not part of the same amplicon. They're 12 megabases apart from each other, and oftentimes there's either gain of the locus in between or just the region in between remains diploid. And we showed, using what is now fairly archaic therapy for neuroblastoma, that in the entire cohort of 2,000 patients, so all risk groups, the presence of an ALK mutation or amplification was a biomarker of inferior outcome. And the same was true when we just honed in on the patients with high-risk disease. And so at the time, in about 2008, we predicted that 14% of patients with high-risk disease these are the patients, of course, with the worst outcome, harbored an activating ALK alteration and could potentially benefit from ALK inhibition strategies. So the very first ALK inhibitor developed was actually developed to target MET, but then was found to be active in patients with non-small cell lung cancer, just as the SOTA paper came out and made the discovery of the ALK fusions in these in a subset of non-small cell lung cancer. So we got this drug in the lab. We called it 1066 for a very long time until it became known as crizotinib. And the first thing we did was just basically treat a bunch of cell lines that we had then sequenced. And what we could see just as a first observation is cell lines that are ALK wild type have no response to ALK inhibition with this drug 1066, but those with an ALK mutation, there is growth inhibition. And then we honed in on the 4 cell lines that represent the 3 hotspot mutations that we see predominantly in patient tumors, as well as amplification. And what we started to recognize pretty early on was that there was a differential response to crizotinib. So that tumors harboring a mutation at residue 1275 and tumors with ALK amplification were exquisitely sensitive to crizotinib, whereas tumors or cell lines with mutations at 1245 and 1174, the second and third most common hotspot mutations, were less sensitive. And we can see this even at the immunoblotting level, where in the more sensitive model, we can abrogate phospho-ALK with just 33 nanomolar of crizotinib, whereas in the more resistant model, we needed higher concentrations of drug to start to see abrogation of ALK activation. We showed the same thing in vivo, so I won't belabor this old data that was published, but I will just say that this was striking for us. These are xenografts that have different ALK mutations, and we had never seen this before in our lab. These complete and sustained regressions upon treatment with a drug that you deliver by oral gavage, and showed an on-target effect here. And we observed, as we did in vitro, that although there's a statistically significant difference when you treat a xenograft that harbors a mutation at this residue 1174, we didn't predict that this would be clinically meaningful. And tumors that are ALK wild type showed really no benefit from being treated with crizotinib. But these data led to really provided the preclinical rationale for the Children's Oncology Group study, ADVL0912. And I put here kind of in bold and red a couple of key points. One is that the study opened in August of 2009, and this was exactly 1 year after the 4 Nature publications on the discovery of ALK mutations in neuroblastoma. And I think that this really shifted the paradigm for drug development. And I owe this accomplishment to my colleagues in the phase 1 consortium who worked with me to push this protocol forward, and the commitment of Pfizer to provide drug to allow us to study this drug before it had even received FDA approval for an adult indication. And ultimately, we defined a recommended phase 2 dose that, this number is kind of meaningless, but it is 200% of the adult maximum tolerated dose. And the reason for that really is highlighted here, and I think speaks to the importance of both the forward and reverse translation that many of us have the opportunity to do as physician scientists. And so as the study started and as we were dose escalating, the crystal structure of the ALK tyrosine kinase domain was solved by 2 drug companies and revealed these pretty unexpected similarities with the tyrosine kinase domain of EGFR. And this provided an understanding for us to predict what the consequences of ALK mutations would be. And I'll just focus on this mutation here at residue 1174 because we knew already that the one at 1275 was sensitive to crizotinib and that it was positioned here in the activation loop. But this residue 1174 lies at the C terminus of the alpha C helix, and it contributes to this very small, kind of well-packed hydrophobic core, and it sits between the alpha C helix and the activation loop. And we know that when it's mutated, say from a phenylalanine to a leucine, that this reduction in the size of the side chain disrupts the packing and weakens the autoinhibitory interactions and allows the ALK kinase domain to adopt an active configuration. So this was the first thing that we started to understand about this particular residue. But more importantly, this residue comes in direct contact, these van der Waals, biochemical forces that I've learned a lot more about ever since I've taught myself more about biochemistry. And this interaction here is crucial because the DFG motif coordinates the transfer of magnesium ions that are involved in ATP binding. And when you reduce the size of the side chain here, this removes the restraint on the position of the DFG motif. And what this does is that it creates an increased affinity for binding to ATP. And so the model is kind of here where altering the DFG motif position allows for this competition between the inhibitor and affinity to ATP. But what we learned about the only ALK inhibitor that we had available to us at the time is that crizotinib binding is not affected by a mutation at this residue. And so we postulated that if we could overcome the system by exposing patients to the highest possible amounts of drug that we could overcome the preferential binding to ATP or affinity for ATP. And that was really the rationale for why we went to dose level 6, as Dr. Fox can speak to, on the trial to really try to get to those really high levels. The results of the trial are pretty impressive, but largely in the patients who harbor ALK fusions. And I think that, Tom Look and Steve Morris are very proud of where this work has led. And actually, Tom calls it the 20-year overnight success story. This is a patient with widely metastatic relapsed anaplastic large cell lymphoma on a PET scan, pre-cycle 1 of crizotinib, enrolled on the study. Post-cycle 1, complete resolution of all metabolically active disease. And what's more important than the initial response is that these are durable. We enrolled 26 patients. This is, what the kind of swimmer's plots look like. This is all already published. What I want to highlight is that this study, designed in 2008, that was open to enrollment in 2009, initially led to FDA breakthrough designation for pediatric patients with ALK-positive ALCL in 2018, and ultimately led to a supplemental new drug application in November of 2020. And while you see some of these patients who came off early, it was because they went into a complete response. And at the choice of the treating physician, they then went on to either autologous or allogeneic stem cell transplant, which for a lot of lymphoma experts still is thought to be the only known curative therapy. Similarly, for patients with IMTs harboring ALK fusions, we enrolled 14 such patients. This is one example of a little boy who had a lot of disease, shown here by PET, in the lung, that resolved completely. And again, the data from this study that was not designed to be a registration study, from which we learned a lot, led to, again, adding IMT to the label in January of this year. So we're very proud of the outcomes of this trial. But the story for neuroblastoma is quite different and really highlights for us the challenges of targeting full-length ALK with point mutations in the kinase domain versus targeting ALK fusions. And shown here are the swimmer's plots for the responses in the 20 patients enrolled on the crizotinib COG phase 1 trial. Well, this is actually the phase 2 data. These are all patients treated at the recommended phase 2 dose. In red are the patients whose tumors had a mutation at residue 1275. These are the patients, if you recall, that we would have expected to have a response. And these were the only patients where there was some hint of activity here, either sustained stable disease or some partial responses and one complete response. But many patients harboring this mutation did not respond, and all other patients with the mutations or amplification even did not respond to this therapy. With one exception, and the exceptions are always important because I think they continue to remind us that you're still on the right track. And we met a little girl who was about 18 months old, and she was born with neuroblastoma, bilateral adrenal tumors, found to also have trilateral tumors, a tumor in her posterior mediastinum. By the time we met her, her disease was pretty refractory to therapy. She had had a lot of chemo, several surgeries, and she was the first child to young parents with no family history at all of cancer. Interestingly, because we learned that she was born with bilateral adrenal tumors, we decided to send her blood to our clinical diagnostic lab, where we had turned our research assay into a clinical assay. We were the first lab to look for ALK mutations and be able to do so in a CLIA-CAP lab. And she was the first patient where we identified a germline ALK mutation, despite the lack of a family history. It turned out when we tested her parents that the mother was at the time a silent carrier. We enrolled her on the phase 1 trial rather than offering her family therapy for high-risk disease, which would have been the next kind of natural recommendation had we not known about the ALK mutation in her germline. And her disease responded beautifully. She had a complete response after 3 cycles. And she took crizotinib for 5 years. And actually, as I was saying to Martine, Thursdays are my clinic day. So I'm in between seeing patients right now. And I just saw her. And she comes for whole body MRIs every 6 months. She's been off therapy for 6 years. She's about to turn 13. And she has been free of disease. But knows that she is at lifelong risk for developing neuroblastoma. And I say lifelong because her mother developed neuroblastoma while she was pregnant with their son, who did not inherit the mutant allele. And we then treated the mom as well with crizotinib, and she went into a complete response and has been in a remission off therapy now for 4 years. So I think that patients with germline ALK mutations, regardless of age, are at lifelong risk for developing the disease. And in fact, the patient I'll be seeing this afternoon is 44 years old and has a germline ALK mutation and continues to have active disease. So in parallel to the ongoing trial being run in the COG, we were studying the functional and biochemical relevance of all of these mutations identified from patient tumors. And we did so using biochemical assays as well as kind of more classic transformation assays. And what we learned from these experiments is that there is differential kinase activation. So first of all, not all of the clinically observed ALK mutations are functionally relevant. All of the ones here that lie in the C lobe of the protein are not activating, at least not to the best of our ability to prove that they are. And those that lie in the alpha C helix in the activation loop are activating, but far less so than those that lie in the phenylalanine core. So we think we now have a way to predict, based on the position of the mutation, whether a mutation or a variant in ALK, as we do more sequencing and as these come up, is going to be activating or not. And as I've shown you, there's differential sensitivity to direct ALK kinase inhibition. Despite that, crizotinib was the only drug we had at the time, and we really wanted to move this forward. And so we decided to see what it would look like when we combined it with standard of care. And these data are in vivo testing of crizotinib in combination with topotecan and cyclophosphamide in 3 models, each that represent the 3 hotspot mutations that we see in ALK in patient tumors. And I'll just focus not on the Kaplan-Meier curve so much, but on the tumor responses here, and just orient you that black are the tumors randomized to being treated by vehicle. In purple, these are the tumors treated just like we treat patients, 5 days of topotecan and cyclophosphamide every 3 weeks. And you can see for each model that at some point the tumors break through and become resistant to the chemotherapy. And they do so, for example, in this patient-derived xenograft, the tumors become resistant to chemotherapy very early on. In red is monotherapy with crizotinib. And this was the first time that in a model that harbored what we thought to be a mutation that was sensitive to crizotinib, we saw that as we continued to treat past 4 weeks, that the tumors develop resistance despite continued therapy. And so we started to recognize that while crizotinib was the first available ALK inhibitor, it was still a weak ALK inhibitor, but in combination with chemotherapy, more importantly, in the models that we knew to have de novo resistance to crizotinib, shown in red here, when combined with chemotherapy, we saw a dramatic synergistic effect, and we showed and published that this is due to a profound upregulation of p53 and massive apoptosis. And these data served as the preclinical rationale for the current COG phase 3 trial, ANBL 1531, which was activated in the spring of 2018. And in this study, all tumors undergo Sanger sequencing of the kinase domain of ALK. And when an activating ALK mutation is identified, patients are non-randomly assigned to Arm E, where they get what is our standard of care, which is a little over 18 months of pretty dose-intensive therapy, including 2 stem cell transplants, and they get crizotinib throughout their therapy. In parallel to all of this work, we were determined to find a better ALK inhibitor, and we were able to publish the comparison of the activity of crizotinib versus Pfizer's 3rd generation ALK inhibitor, now known as lorlatinib. But, you know, the dirty little secret is, and I know we all do this in our labs, is that we had also looked at all of the other available ALK inhibitors being developed by our industry partners for patients with non-small cell lung cancers who were developing point mutations in ALK as a mechanism of resistance. So, you know, you can easily see in the biochemical assays and in the cellular-based assays that the drug 3922 or lorlatinib, first of all, it doesn't differentiate between the mutations. And so it's significantly and equally potent against all of the 3 hotspot mutations in the biochemical assays at nanomolar concentrations that are much lower than crizotinib. And then the same was true when we did the experiment in NIH3T3 cells. We took this drug into our in vivo patient-derived xenograft models. And in panels ABC here are models that harbor the crizotinib de novo resistant mutations. So tumors randomized to vehicle are in black. In blue is what happens when you treat with crizotinib, and in red is what happens when you treat with lorlatinib. And so these data for us were super exciting. We had not seen this with any of the other ALK inhibitors. And we also saw that in the model that harbored the mutation that is initially sensitive to crizotinib, here shown in blue, that the response to lorlatinib was sustained. And using a chemical proteomics assay that I'll talk a little bit more about, we really did show what seems obvious, but we proved that, you know, we were able to demonstrate with data that lorlatinib is significantly more potent in turning off the active state of ALK than crizotinib is. Other work that we've done to try to enhance the activity of lorlatinib is also combined it with standard of care chemotherapy. So you take this model here, this patient-derived xenograft model that has a mutation at a crizotinib-resistant residue 1174 and has MYC amplification. And these are in green tumors treated with topotecan and cyclophosphamide. So this is a pretty chemotherapy-resistant model. We learned here that lorlatinib, while seemed like a home run, is a very potent ALK inhibitor. But after 10 weeks, the tumors, despite continued therapy, break through and develop resistance. But when combined with chemotherapy, we were able to see continued response. The arrow is a symbol for when we stop therapy. And these tumors do eventually grow back, not in all of our models, but in this particular model, it takes about 12 weeks or so before the tumors start to grow back. What was really exciting to us is the data here, and I'll show you why a little bit later, but this is the only model that we have that harbors not only MYC amplification, but high-level ALK amplification. And both the Europeans and the North American consortia have shown that patients with these two alterations have the worst outcome when diagnosed with high-risk neuroblastoma. And you can see the effect of lorlatinib alone is superimposed on the effect of lorlatinib with chemotherapy, whereas chemotherapy alone is effective until we stop chemotherapy and then the tumors become resistant. When we stopped lorlatinib and stopped the combination of lorlatinib with chemotherapy, actually these tumors have never regrown and have been sacrificed now because they're over, they started becoming geriatric mice. So this was really exciting for us. And our lab has been really interested in understanding why lorlatinib is so superior to crizotinib. And it may seem obvious, and it may just be that it's far more potent in inhibiting ALK. But we collaborated with Gary Johnson at UNC, who's the head of pharmacology there, using his multiplexed inhibitor beads platform to enrich for active kinases. And we coupled that with quantitative mass spectrometry. And what I show you here is basically the kinome reprogramming of patient-derived xenografts with an ALK mutation treated either with crizotinib or with lorlatinib, and tumors collected at 6 days. And what the state of the active kinome looks like. And I think, it's obvious in these volcano plots that crizotinib has very little effect on the kinome. And I can't really see this arrow here, but this is where ALK is. ALK is initially downregulated and inhibited, but it actually rebounds quite early on. And so it's only partially abrogated in terms of its active state. And then it despite continued therapy, it rebounds. Whereas with lorlatinib, ALK inhibition is profound and sustained. But even more striking, or I should say as striking as that, is we saw this downregulation of all of these other kinases, many of which are involved in the transition in the cell cycle between the G2 state and the M phase of the cell cycle. We saw very few active kinases that were upregulated upon treatment with lorlatinib. So this starting to shed some insights into the differences between lorlatinib, crizotinib, and the rationale for doing this experiment for us is to see if we can identify other pathways to co-target with lorlatinib to continue to improve upon the anti-tumor and anti-ALK activity of lorlatinib. This is the exact same experiment, except done using RNA sequencing. And again, I'll just sort of point your attention to the RNA sequencing data here for a model that has a crizotinib-resistant mutation treated with lorlatinib. and we see downregulation at the transcriptome at the transcript level of the G2M kinases. And interestingly, we see upregulation of the TRAC kinases, which is interesting because Pfizer has always thought that these were actually downregulated by lorlatinib. So this is data for us to ponder, and I can elaborate on why that is later, and directly related to some of the side effects that we're seeing in the clinic with this drug. But these data were the preclinical rationale for the phase 1 trial of lorlatinib that opened in the NANT in 2017. And this trial is now completed, although it remains open to enrollment so that patients can continue to have access to the drug. And we decided to have 2 separate cohorts, one for pediatric patients and one for adult patients, because we know that adults, actually there's a higher frequency of ALK mutations in adults. We don't really understand why that is. For the pediatric patients, we've defined a recommended phase 2 dose that, again, is 200% of the adult recommended phase 2 dose. The adults never really hit an MTD, so it is called an RP2D. And for the adults, we're defining a dose that is above the standard dose used for patients with non-small cell lung cancer. As part of this trial, we've had the privilege of performing serial ctDNA assays throughout the course of therapy. And I think this is really one of the first trials, biomarker-driven trials, with a targeted therapy where serial liquid biopsies are being performed. And this is giving us some insights into mechanisms of resistance. But I want to just show you very briefly the rationale for why we continue to push the dose of lorlatinib, just like we did with crizotinib on the COG trial, which is that when we dosed our PDXs with these resistant hotspot mutations at a lower dose, this is equivalent to the dose used for non-small cell lung cancer, we saw really a cytostatic effect. At a higher dose, we saw really complete, sustained anti-tumor activity. Interestingly, in non-small cell lung cancer, when you have the native ALK fusion, EML4 ALK, you only need, I know it's hard to see in green, 3 mg per kg to achieve anti-tumor activity that is really robust. And that led to FDA approval of lorlatinib. But when patients with non-small cell lung cancer have an EML4 ALK fusion and develop, or I should say acquire a mutation as in the ALK kinase domain as a mechanism of resistance, and here it's the L1196 mutation, this is the gatekeeper mutation, there you only see responses when you go up to 10 mg per kg. So we felt that this provided the rationale for us to continue to increase the dose. And these are the kinds of responses that we're seeing on the trial. We're learning a lot more about how targeted therapies work. So this is a patient with a large mass primary refractory neuroblastoma that is in the right carotid space. And you see the very bright MIBG uptake that after 4 cycles starts to really significantly decrease in its uptake. And by course 10 was called negative. And this patient has been on lorlatinib for 4 years. We actually went in and biopsied the remaining tissue, which by RECIST criteria has not changed in size. And all we see is either scar tissue or differentiated ganglioneuroma. Interestingly, based on the criteria that we use now, this patient is going to be called stable disease. But I think it's really important for us to really understand how targeted therapies work. This is another example of a patient treated on the adult cohort who was heavily pretreated with prior ALK inhibitors, who had not only an ALK mutation, but had acquired a P53 mutation in addition to these other mutations, and had, as you can see on this MIBG scan, disease throughout the entire appendicular skeleton. She benefited from this therapy, had a complete response for 2.5 years, and unfortunately died after suffering a relapse. She was 45 and had 3 kids. the serial ctDNA that we're doing is providing insights into mechanisms of resistance. And what we're seeing is that those are mainly coming out at the lower dose levels. So this is an early patient. You can see here on this axis, the y-axis is the variant allele frequency, and the x-axis is time in months of when we access these samples and subject them to sequencing in collaboration with Foundation Medicine. You can see that the parental ALK mutation, which is ALK 12 at the residue 1245, the variant allele frequency comes down by 2 months, and then as the patient then progresses through therapy, it starts to rise again. And when we hone in on what's happening here at much lower variant allele frequencies, we start to see the acquisition of new mutations, not only in genes like PTPN11 and NF1, but also new mutations in the ALK kinase domain. So this is at least a mechanism of resistance to therapy with lorlatinib, and this is a major focus in the laboratory right now. All of this work has led to Amendment 8 of ANBL 1531, which I'm proud to say was fully approved just last month. And that is exactly 14 years after the discovery of ALK mutations in neuroblastoma. And in collaboration with our European colleagues, we are integrating now lorlatinib upfront. So for the COG trial, it means that we are replacing crizotinib with lorlatinib. We have a completely new analytical plan for this arm of the trial. Because of the absence of hematologic toxicities with lorlatinib, and because of the mechanism of action of the drug, we don't expect any overlapping toxicities. And we're finally in the COG moving away from Sanger sequencing, and we developed a next-gen sequencing assay in my laboratory that's now been CLIA CAP approved in Elaine Martis's lab centrally for all patients who will be enrolling on this trial. And that will start to happen this month, that patients will be transitioned to lorlatinib. We were asked by the FDA to have a proper control arm if we wanted this to be a registration study. And the only way to do so is really to compare apples to apples, right? So to compare patients with ALK mutations getting lorlatinib to patients who with ALK mutations who did not get lorlatinib, but who also got contemporary therapy. And the only way we could think of doing that was sequencing tumors, available DNA from available patient, tumors from the predecessor study, ANBL 0532. These are new, unpublished data. We sequenced 250 some tumors. That's all that we could get from this study. But the data, I think, speaks for itself. Patients who are ALK wild type have a 5-year event-free survival of 51% versus those with an ALK aberration have a 5-year EFS of 34%. So this will serve as the control for the current trial when lorlatinib gets integrated. What I also want to highlight is now with NGS, we can show that the frequency of ALK mutations in diagnostic tumors is far higher than that original number. I quoted you of 14%. It's really 22%. Not all are clonal. 12% are clonal, and then the rest are subclonal, and the variant allele frequency can really range. And we've had the opportunity with our next-gen sequencing assay to also define the landscape of the RAS pathway mutations in diagnostic tumors, which is non-trivial. It's close to about 8%. And we're further exploring the prognostic implications of this. We know that ALK mutations at relapse are much more prevalent, and we have shown and not yet published and have a collaboration that's ongoing with Jingwei and Sam Brady at St. Jude to look at this. But when we sequence all our PDX models derived from patients at relapse, the frequency of ALK mutations is close to 40%. This is the pie chart that shows kind of the rest of the breakdown of other mutations. And we know that these mutations aren't necessarily present at diagnosis, or maybe they are and we just don't have the technology yet the ultra deep sequencing that we need to detect them. But a patient here, no ALK mutation, this is their therapy over, starting from day 0 all the way until time of death. And the ALK mutation becomes detectable only much later on in therapy. And this really, I think, provides the rationale for either serial liquid biopsies or for tissue lesional biopsies at time of relapse. So we're tired of using the Foundation Medicine ctDNA assay because it wasn't developed for pediatric childhood cancers specifically. So we're developing our own ctDNA assay. Right now, it's pretty neuroblastoma specific, but our goal is to make this a childhood cancer ctDNA assay. And I show you here our ability to detect ATRX deletions as well as ALK amplifications. And this current assay has about 25 genes on it. So now I'll transition to ALK as a promising immunotherapeutic target. I've been talking a lot about ALK in its mutant form with point mutations in the kinase domain that lead to autoactivation. But we know that native ALK is expressed on the cell surface of the vast majority of all neuroblastomas, and that on this pediatric normal tissue microarray that we built at CHOP, there's essentially no expression. So the expression of native ALK is restricted to tumor. And we think that for that reason, it makes this a really promising immunotherapeutic target. So we panned for antibodies, antagonistic antibodies against ALK. I guess that's sort of redundantly said. And we eventually made an antibody drug conjugate and showed that it is very active in cells that have ALK expressed on the cell surface, even cells that are ALK wild type. That was the purpose of this work, is to expand the role of targeting ALK, that ALK wild type can be targeted since the extracellular domain is intact and is expressed on the cell surface. What we see is that response differs based on the amount of the density of the receptor on the cell surface. And we took the in vitro work with our ALK antibody drug conjugate, took it in vivo, gave 2 doses in models that are ALK wild type but have ALK on the cell surface, and showed that there is anti-tumor activity. So this was early proof of concept. And where we're going with this work is we've humanized the antibody. We're looking at different toxins, PBD and duocarmycin, and finding that our lead ADC is what we call ADC6, which happens to be conjugated to PBD. And you can see that it is very active against models that have all different levels of ALK on the cell surface. And this is our negative control, no ALK at all on the cell surface. And we really see, only activity at high doses, which is probably due to, at this point, free drug. So we're really excited about the concept of ALK immunotherapy. And some of you may have seen that the extracellular domain of ALK, the crystal structure, was just solved again simultaneously by 3 groups and published in Nature. One of them are our collaborators at Yale, and they defined this glycine-rich domain, which lies right here, just proximal to the transmembrane. So it's really membrane proximal, and this is the region where there is ligand binding and dimerization. So we think it's key that in the development of antibodies for developing cell therapies or other immune therapies to target ALK, that the antibodies be targeted to this region. So this has been very informative, and we've been able to pan for antibodies like this one, VH20, which binds to the glycine-rich domain, as shown in these ELISA assays, and has really nice signal here in cells that I show you here the data that comparing VH20, our newest antibody, to our humanized antibody that we used previously to develop an ADC. And now we're using this particular antibody to develop a bispecific T-cell engager, so targeting ALK and targeting CD3, and show activity in 293 293 T cells with forced overexpression of ALK. And I won't go into these because I know I'm running a little bit late, but it is a very promising approach. What's also exciting is that native ALK is expressed on the cell surface of the majority of fusion-positive alveolar rhabdomyosarcomas. And so we're working with Peter Houghton to test our ALK ADCs and BiTEs to see if this could be a therapeutic approach for this other, very lethal disease. Another target that we're going after in collaboration with John Maris's group and Lou Chesler and Emily Bernstein is ATRX, and this also comes from St. Jude, right? It was your team, Mike Dyer, and collaboration with the folks at Sloan Kettering that showed that ATRX alterations really define indolent neuroblastoma. And so we're really going after this by developing assays not only for detecting ATRX alterations, but to try to define what is the biomarker of indolent neuroblastoma. Is it just ATRX or alterations, or are there tumors that have what we call, this alternative length telomere phenotype that can be assayed by this C-circle assay to define the indolent phenotype? We're developing preclinical models, and Lou Chesler is doing that and testing rational therapies. And the one that looks the most promising right now is work that Emily Bernstein showed that the PRC2 complex is integral in the context of an ATRX, either in-frame fusion or alteration. And we're working with Pfizer's new drug. And what you can see here is that at very low doses of Pfizer's EZH2 inhibitor, we can really start to turn off the key H3K27 trimethyl mark, whereas these other drugs, one of which is tazemetostat, you need micromolar doses. And this is some early data comparing tazemetostat to the Pfizer drug in models that have either ATRX in-frame fusions, showing that this drug is very potent. And similarly, it's very potent in models that are ATRX wild type, this is misspelled, but have an alt phenotype as defined by C-circle assay. These are tough experiments because you have to really take them out for a long time, but by 28 days, we see really nice activity at low nanomolar concentrations. And then the last project that I'll just very briefly mention, which I'm most proud of taking on, at this time in my career, is really drugging MICN. This is our all-star team, and I'm so proud to be working with Martine, really. I think, the MIC guru. And we have a phenomenal team as well as a collaborator with a pharmaceutical company that is really engaged. And they are really committed to developing degrader approaches. And the reason why we're going after MICN may seem obvious. I'm not going to go into Felix's story here, which is so important to my own motivation for going after this. But what I want to just show you is that MICN, seen all in red, is always a truncal event in neuroblastoma. ALK can be truncal, especially when it's a germline mutation, but it's often a branched event. It's subclonal, or it's only seen as in a subset of cells. But when MICN is amplified, it is a stable alteration, and it's always at the trunk. And in this patient, and in work that we've done with Jingwei and Sam, we see, the ALK mutation throughout different modalities that we've used to sequence it. And there's always MICN that I don't show. We're able to treat ALK, we were able to get away from the ALK problem with lorlatinib. Then NF1 came up as a branched event, but mostly it's all about MICN. And so we think that by taking an approach with a new category of medicines using these protein degradation drugs, some of us call these degraders or PROTACs, that we can finally go after MICN. And MICN has been considered undruggable because it's largely unstructured. It's really the c-MYC domain that is more structured, but the domain where MICN resides is really actually plastic and only becomes structured when it's bound to another protein. And this allows for perhaps an interface that could become druggable. And I'll leave you with one last slide, which is hopefully a movie that will play, where this gives you the impression of how we envision the plastic region of MICN becoming structured. And so what this shows you is that MICN lacks structure and solution in the absence of a binding partner. But when it comes into contact here specifically, as Richard Bayless's group showed, with Aurora A, or it could be another binding partner, it becomes structured on the surface. And so we're really going after this. And Martine and I are proud to say that just last week, we received our first set of degraders to test. So we're really excited about this. And I hope in the future, I'll have the privilege to be invited back to talk about this project. So I apologize for going a little bit over time, but I really want to thank the many folks in my lab, my collaborators, both at CHOP, collaborators at other sites, funding, and of course, the patients who really inspire my work. And thank you very much for your time.
Thank you so much for a wonderful talk. So I know we are at the top over the hour. First, I would like to thank Dr. Murthy for her talk. We'll close the official event, but there's several questions on the chat. And so for those who would like to stay, Yael agreed to stay with us and answer questions. So the first is from Camila Hashimoto. She says, I have a 7-year-old girl with ethnic amplification and ALK F1174C mutation, refractory high-risk neuroblastoma. She has now disease progression, lung and bones, for the third time, and is already taking lorlatinib at 95 mg per meter square for 6 months now. And she showed response to lorlatinib in the first 2 months. I repeated the tumor biopsy and liquid biopsy last month with the same ALK mutation, ALK F1174C, blood VAF 0.41% in Foundation 1, and she already undergone standard SCT and chemoimmunotherapy. And I'm planning MIBG with vorinostat as a trial with CAR-GD2 in Rome. And she said that the trial with CAR-GD2 in Rome is closed. So she has grade 4 hematologic toxicity. And what would you recommend regarding ALK inhibitor? Increasing the dose of lorlatinib, changing ALK inhibitors? So it's of course hard to give advice about patients, but what I can tell you in our experience, both in the clinic and in the laboratory, is that even when these tumors become resistant to lorlatinib or start to develop and show resistance, if you relieve the pressure of the ALK inhibition, the tumors explode. And this has been seen in non-small cell lung cancer. We see the same thing in neuroblastoma. And so there is a retained partial dependency or partial oncogene addiction. And so my approach is never to stop the lorlatinib. Unfortunately, it's tough if you're trying to enroll a patient onto a clinical trial, because in that situation, you have to stop the lorlatinib. But, in a situation like this, just based on the summary of the patient, I would continue the lorlatinib, increase to the 115. And we have, now our MIBG protocol allows us to treat with vorinostat and lorlatinib because we know that these patients will suffer rapid disease progression if the lorlatinib is discontinued. So and we've also shown that it's safe to give with chemoimmunotherapy if the patient did respond to chemoimmunotherapy. We're really looking for novel combinations and partners to lorlatinib. And we have some hints that combining lorlatinib with aurora kinase A inhibition is going to be highly synergistic. Unfortunately, that field has really failed for adults. And so we're really hoping that we can develop that for pediatric ALK-driven neuroblastomas. That would be my recommendation, is to keep the lorlatinib on, even in the context of progression, raise the dose and integrate it with MIBG and vorinostat. Thank you. Charlie Wright, thanks for the wonderful talk. Have you tried combination of lorlatinib with any other standard of care drug? besides cyclophosphamide and topotecan? Could some effective combination decrease the lorlatinib dose to maybe mitigate the MTD problem?
Yeah, that's a great question, Charlie. And I think one of the challenges in testing the other standard of care drugs is that they're so toxic in mice, platinum, doxorubicin. So we have done that. And what we'd like to do is actually provide the rationale for decreasing the dose of the cytotoxic agents, not the dose of the lorlatinib. I mean, lorlatinib is extremely well tolerated, and at least in our experience, has very few side effects. Of course, we don't know what the long-term side effects are going to be, but I think that our goal should be to decrease the intensity of our very empiric backbone of therapy.
Thank you. Diane Acevedo, do you know if there's any effect of targeting ALK in neuroblastoma cell population, i.e. mesenchymal versus adrenal?
Yeah, that's a hot topic, right? This adrenergic mesenchymal state switch. And it's really difficult to study in the lab. And the reason for that is that all models are models, and therefore all models are wrong. And the experience can only be from what happens in patients. We are able to isolate mesenchymal cells from patients, and those lose expression of ALK. And so clearly ALK is an adrenergic marker. Most of our PDXs do not have even subclonal mutations in ALK. They're all highly adrenergic. And so it's really a tough thing for us to study, but it is something that I think is really integral. I think that these are what we call persister cells. I think they can be called many different things. Are they the cells that are adapt to our therapy and have this state switch? I think they lose ALK expression, just like they lose FOX2B expression, like they lose a lot of the adrenergic markers that define the core regulatory circuitry. And I think these are important to understand in developing co-targeting strategies.
Thank you. Adam says, phenomenal work, Yael. Camila Hashimoto, thank you. I have a question about the toxicity. Could you address, you said it's well tolerated, but we know that, it can be just like water. So I would like to know what, are the toxicity. And then the other question is, why do you think children tolerate super high dose compared to adults? Is it because of the radiation burden, or is it something else?
I think that that's true for most of the work that's been done in the phase 1 consortium and in other phase 1 studies, that we find that children have, despite the fact that they've been exposed to so many cytotoxic agents, they're less exposed to some of the environmental factors, and they're just able to tolerate much higher doses of drugs than adults are. And that's been true for at least a lot of the targeted therapies trials that I've been involved with. Lorlatinib and crizotinib are also really interesting because they do have a nice side effect profile. Well, I should say it is a significant side effect profile, but it mostly impacts adults, and kids actually have very few side effects. The main side effect is significant weight gain and hyperlipidemia that can easily be treated with statins, although the vast majority of kids don't need statins. Their cholesterol can be managed just by diet alone or fish oil. And kids don't get the side effects that the adults do in terms of the peripheral edema and the peripheral neuropathy. The weight gain is attributed to the same reason as the main side effect of this drug, which is the neurocognitive adverse events that are seen in adults. And there, it causes short-term memory loss, issues with word finding, and changes in lability with mood. And at least Pfizer's explanation for that is that It is highly, it crosses the blood-brain barrier quite potently, which is a wonderful thing. And we're seeing some incredible activity in the infants with high-grade gliomas getting lorlatinib, which is really exciting. Pfizer has shown that a third of the levels seen in the overall are in the CSF. And they attribute it to the impact on TRAC. And they think that it's downregulation of the TRAC receptors that causes patients to lose their ability to control their appetite. And they also think that that's the reason why the effect on TRAC causes some of these CNS effects. We're seeing upregulation of TRAC. So we have to kind of reconcile how TRAC is playing a role in the side effect profile. But we see very few neurocognitive side effects in children. And the weight gain and increase in appetite for these kids who will now be getting this upfront, we think will be only a benefit to them.
Thank you. So I have one more question, and then I'll let you go. And that is that it looks like lorlatinib, based on your mass spec, is actually, affecting many kinases compared to the other drugs, right? That seems to be more specific. So you think this is why this is so potent?
I do, Martine, and we have a manuscript that's under review that will hopefully be published soon. That we think that it's not just because lorlatinib is a potent and specific ALK inhibitor, but because it really potently downregulates these G2M kinases. And that was not shown by Pfizer originally in their first biochemistry paper and their enzymatic assays. They did not see this. They saw downregulation of TRAC and ROS, but they did not see any impact on the kinases involved in the cell cycle. So we do think that this is a mechanism by which that lorlatinib is so far superior to crizotinib.
Thank you. And then one last question that just came in. Can you tell us a bit more of the response in patients with CNS relapse or CNS disease?
Yeah, we're seeing dramatic responses, and they're sustained. There have been a couple actually single case reports published, and it's really exciting, right? This is of course a major problem in the field for all of us, and for neuroblastoma, a lot of patients are suffering relapses in this sanctuary site where the majority of our drugs don't penetrate. We know that GD2 as a antibody doesn't get through the blood-brain barrier, so. Yes, I've seen responses not only in neuroblastomas with CNS relapses, but anaplastic large cell lymphoma with leptomeningeal disease in infants with high-grade gliomas. And so I think that this is one characteristic of this drug that also makes it even more attractive.
That's wonderful. Well, thank you so much. It was wonderful to see you. Always, yeah.
Thank you, Charles. Thank you, everyone. Yeah, and I'll see you on Monday, Martine.
Thank you very much for staying around and being the Q&A. And I'll let you go, Yael, to see your patients. Thank you so much.
Thank you also very much. Have a wonderful rest of the week and weekend, and be safe.
Yes, too. Bye-bye.